Good morning, everyone. Welcome to the Hardwood Biofuels webinar series. Today we will have Brian Stanton from GreenWood Resources and David Neal from University of California, Davis. They will be talking on utilizing hybridization tools in Poplar for bioenergy. And I see we primarily have an academic audience with us today, as well as a few people from extension and business. It seems that most of you are highly familiar with breeding poplar for bioenergy, and so this will be an educated audience for Brian and David. Having a bit of trouble here moving to the next slide I wanted to show you, so just hang on for a minute and your patience is appreciated. Okay, so just before we get started, I want to just give you a bit of background on the project Advanced Hardwood Biofuels Northwest. The research that Brian and David will be presenting today is part of this project. This project is a 5-year grant from the United States Department of Agriculture. It focuses on 5 different teams to look at all aspects of growing poplar and then converting it into biochemicals and drop-in replacement transportation fuels. The project is a consortium of university and academic university partners and industry partners. It's led by the University of Washington, and then we also have University of California, Davis, GreenWood Resources, and they will be doing the webinar today. So now I'd like to turn it over to Brian Stanton from GreenWood Resources. He's going to be speaking first, and then after he speaks, it will be David Neal from University of California, Davis, and they will be speaking on using molecular tools in poplar hybridization for bioenergy. If you notice, there's a chat box on the left side of the screen, so if you have questions, please type them in there, and both Brian and David will have time at the end of today's webinar to address your questions. Thank you.
Patricia, thank you. This is Brian Stanton. Good morning. I'm with GreenWood Resources. I'm the Chief Science Officer here. But this morning I'm speaking to you as the lead of the AHB feedstock production team. This webinar this morning is going to cover 4 topics. First, I will give a very brief overview, not so much of the AHB project as shown on the screen, but more the feedstock program that operates within AHB. This feedstock program, it's designed to improve the yield, the quality, and the cost of growing hybrid poplar biomass. Part of that research involves hybridization and varietal selection. So after the review, I will next describe the classical approach to poplar genetic improvement that was originally designed by Ronnie Stetler at the University of Washington in the 1980s, but now continues today in the Northwest under GreenWood Resources direction. I'm doing this because it's setting the stage, so to speak, for what it is we will be trying to first accomplish with the application of molecular tools that you will hear about from David. So after the review of the classical approach, I'll turn the presentation over to David for an overview of his work at the University of California, Davis, to develop these genomic-based breeding tools that will augment the classical approach to poplar improvement. And then after David, I'm going to come back to close the webinar with GreenWood's plan as to how these tools are going to be first applied in our operational poplar improvement program. So just a quick word before I get started, a quick word about the AHB geography. You know, the Pacific Northwest, it's often defined as Oregon and Washington, but within the project, the AHB project, we take a broader definition that extends the Northwest eastward into Idaho and western Montana, and then again southward to the coastal region of Northern California. And in this 5-state region, major petroleum refineries are located in the north, Washington's North Puget Sound region, and then going further south into California's Bay Area region, major petroleum refineries. Fuel moves throughout the region in 4 major pipelines, and also by barge up and down the Columbia River, and to a limited extent by ocean transport from California northward. And then in addition, major fuel depots are located at various points on the pipeline network: Spokane, Washington, Portland, Oregon, Eugene, Oregon, Boise, Idaho. And I'm mentioning this because the importance of the network is that at the inception of the project, the AHB project, sites were identified where we, GreenWood, and our conversion partner, Zeachem, envisioned that the commercial potential for the bioenergy plantation operations would first take hold. And that was in consideration of this network and also access to markets. So then hybrid poplar demonstration farms were established within Washington, Oregon, Idaho, and California, again, each in proximity to major population centers or a refinery or a pipeline terminal or a fuel depot. Now, the purpose of the farms is to refine estimates of biomass yields, production costs, exhibit harvesting technology, just showcase the whole poplar cropping system. These farms vary between 20 and 40 hectares in size. They're generally located on alluvial plains and terraces, but one is situated on a Cascade Piedmont location. Terrain is level. One site, the Cascade Piedmont site, is sloped up to 10%. Precipitation runs between 450 and about 1,200 millimeters. Elevation ranges from sea level up to 700 meters. Soils, for the most part, are clay loams, and silty loams. So a diversity of sites to demonstrate the poplar system. Each of the farms then was planted with several highly selected poplar varieties. About 7 to 11 locally adapted hybrids are set out in each of these 4 locations. And then all sites have been harvested following their initial 2-year planted cycle. They have been regenerated by coppice, and they will be cut after the 3rd year of coppice growth as the 5-year AHB project concludes. So just to give a brief introduction to what David will be talking about, the feedstock program includes a number of research projects. Some of these address screening clonal varieties using the traditional approach, screening varieties for coppice biomass production. We're also developing an indirect selection protocol for resistance to herbivory using near-infrared spectroscopy. We're looking at endophytes and how they can apply to increase disease resistance, drought tolerance, confer a measure of nitrogen fixing ability. We're looking at genetic transformation for sterility. And finally, the focus of this morning's webinar, the UC David development of molecular breeding techniques. But before we look at those techniques, we really need to, it deserves mention that the traditional approach to varietal development has long been based on somewhat of an unusual, at the time it started, an unusual domestication strategy. So, to quote the American poplar breeder, Ernst Schreiner, back from 1966, it is the genus through interspecific hybridization that provides the wide diversity of genotypes needed for maximum genetic improvement. Since Schreiner's early work in poplar genetics, the classic approach to poplar breeding has relied on first-generation hybridization as the predominant, the main controlled breeding strategy. It frequently results in heterosis for growth rate. Now, as you advance hybridization into the second interspecific generation, at times you'll recover transgressive segregants or individual genotypes that perform as well or better than the parents. But generally speaking, the average performance of the F2 generation is far less than the F1 generation. It's not commonly pursued. I'll mention that one reason might be a reduction in heterozygotic loci, along with a disruption of species-specific linkage groups or gene complexes during F1 hybrid gametogenesis. The production of pollen and egg cells might explain this diminished vigor in the F2 generation. Okay, and keep that in mind because David and I will be coming back to that. Now, there are three reasons or objectives in first generation hybridization as articulated by Ronnie Stetler 30 years later. Those are to combine desirable traits from two species into one hybrid variety, to capture heterosis or hybrid vigor, or to increase developmental homeostasis. Okay, these three motives, they overlap. They can't always be cleanly separated, and this is partially due to the fact that we really don't have, at the molecular level, a clear understanding of how heterosis or hybrid vigor is inherited. The phenomenon remains somewhat elusive. It presents a roadblock to maximum poplar breeding efficiency. This is a big issue. Genomic tools in the future should help resolve this. Okay, the populist genome, the stuff or the resource for which genomic-based breeding tools are being developed. It's made up of 19 chromosome pairs. This is a relatively large basic chromosome number compared to other dicots, forest trees. And it also may be associated with an increased frequency of recombinant gametes that are formed during reduction division. And what this does, it increases the level of within-family variation, and that is important because it drives clonal selection and propagation. Equally important, I think, is the fact that the poplar, the genus Populus, all species are dioecious. They're characterized by very high seed to pollen ratios. This leads to a tremendous capacity for sexual recombination, contributes to the production, the recovery of genetically variable families. This too is important to advancing hybrid improvement. And again, I'm mentioning this because both levels of genetic variance, both the among family component as well as the within family component, are going to be very important to how we apply genomic tools in GreenWood's hybridization program. Now, as far as the hybrids themselves, or the hybrid combinations, the hybrid taxa that we pursue in the AHB project, there are three. The first is the canadensis taxon, the hybrid of Populus deltoides, eastern cottonwood, and Populus nigra, European black poplar. We are using these in areas of continental climates. Second is the Populus generosa taxon. This is a cross between the Pacific Northwest species Populus trichocarpa and again eastern cottonwood Populus deltoides. And then finally, the third taxon, Populus deltoides with Populus maximowitzii, Japanese poplar from Asia. We're using this in continental climates where the canadensis taxon is most important, and then also in coastal environments where the Populus generosa taxon is used preferentially. Okay, GreenWood's hybridization project, we use parental selections. These are maintained in very, very large breeding arboreta. You see one in the upper left. We presently maintain 5 of these arboreta for the 4 species. each of approximately 150 genotypes. These arboreta are located in the northwest. There's one in the southeast. Selections are crossed in greenhouses using 1.5 meter long cuttings. Pollen is forced in the winter. Pollinations are made in the spring. Seed catkins mature in the same season. So a pedigree population can be produced in 1 year. And we annually breed in greenhouses between 125 to 150 full sib families of those 3 hybrid taxa that I mentioned. Field trialing. Field trialing of the material, it's clonally replicated. You see in the center photograph, a photograph depiction of how poplar is clonally vegetatively propagated from hardwood cuttings. All our tests are clonally replicated in view of the added precision, the efficiency it contributes to genetic selections. But when you are doing this, you can replicate the individual genotype. You create a potential for very, very large test populations. And as a consequence, we're using a multiple stage evaluation process that accommodates such large populations. So in this multiple stage testing protocol, the number of genotypes as it moves through the stages, that the number is sequentially reduced between stages, along with an increase in replication, an increase in spacing, an increase in test locations to account for genotype environment interaction, and then rotation length. Typically, testing begins with large progeny collections that undergo combined family and within family selection. Then the trialing transitions at the final stage to a limited number of highly selected clones that are tested in monoclonal plots. And these are large enough, we feel, to give reliable estimates of growth and yield. Test rotations last 1/3 to 1/2 of a commercial rotation, which, depending on the market that we're selecting varieties to perform within and how much risk we are willing to take, this process can be as brief as 3 years when making selections for the bioenergy market, or as long as 15 years for the sawnwood market. Okay, so that is a very, very brief overview of poplar breeding, reproductive biology, and tree improvement. And I feel it's necessary to do this to place into context the way that GreenWood's tree improvement program is endeavoring to make application of the tools that are being developed. They're not fully developed yet, but the tools that are being developed by David and his...
I hope momentarily.
Patricia, I'm back. Can you tell me at what point of the presentation you lost me?
I think you were on this slide right here, Brian.
Okay, that's good. So this concludes the introduction to poplar hybridization, reproductive biology, the taxa that we're targeting, and the process we're using to advance varietal development for the bioenergy market. So at this point, I'd like to turn the presentation over to David to describe the work that he and his postdoctoral associate, Fernando Guerra, have been doing at the under AHB, have been working at the University of California, Davis to develop genomic-based tools. David?
At this moment, I'm back, if you can hear me.
Very well, David. Proceed.
Yeah, the software was very mean to me. It completely, without me touching a key, just threw me overboard. I had to go all the way back out to the beginning and start over. I don't know whether I'm being heard by the participants or not. You want to confirm that for me? Whether we even have any participants still on the line with all these problems. Patricia, can you confirm that I'm being heard by everyone?
Yeah, I think so. You're loud and clear on my end. So if Brian Stanton and Brian Sarlo could turn off the mic, we'll eliminate some echo if there's just two of us on the line.
Okay, well, I apologize to everyone for these problems, but they were out of my control here in Davis. So yeah, this is David Neal in Department of Plant Science at UC Davis. Just a quick introduction. Brian Stanton and I at GreenWood Resources have been working together on trying to develop molecular-based breeding in poplar for a number of years. We got started some years ago with some funding from Chevron Oil Company, actually, and then a small number of grants from the USDA. But those were sort of small-scale projects, to begin the process, but really couldn't think about taking this to scale and ultimately application until there was larger sources of funding, and that was made possible through the Advanced Hardwood Biofuels Project, the USDA-funded NIFA project. So I will just quickly go through the steps in research and development that are needed before we can even begin application of molecular-based breeding for biofuels. In the first slide here, I give 3 reasons why one would want to move from a phenotypically based or classically based program that Brian has just described to a molecular-based program, and these are the same reasons that are used in any agricultural system. The reduction in time from breeding cycle time that can be afforded through markers versus phenotypic selection, decrease in cost, and increase in precision. But these justifications are actually maybe even more so important in forest trees. That we know the time to evaluation and selection for some traits can be quite lengthy, on the order of a decade or more. So selections can be made at time 0 practically. There's a great cost. In the early days, it was argued that the cost of molecular breeding was much too expensive relative to phenotypic selection, but we've seen over time that those costs have decreased significantly, but the cost of land and fuel and labor and so forth continues to increase. So the cost of molecular breeding is now probably somewhat less expensive than phenotypically based programs. So in the next slide then, I will just go through the basic steps that one needs to take in order to begin the development of a molecular breeding program. And one has to begin with a population. Yes, you'll see in the upper left-hand corner what's called the association population, where in a population of trees, we begin to understand the relationship between the phenotype and the genotype. The population that we have used is a large clonal population of Populus trichocarpa that was assembled by Brian and his team at GreenWood. These trees, initially these clones were planted in plantations up in his region in Washington and Oregon. It was approximately 460 clones that were more or less genetically unrelated to one another, which is sort of optimal for identifying marker trait associations. With the funding from the most recent NIFA project, we established a replication of the same clonal population here in Davis, and that's where this picture is taken. So it's the same 460 clones, 3 reps each, and a large clonal population here in Davis. So now we go about phenotyping and genotyping that population. You'll see in the next panel there below the association population, the section entitled phenotyping, the different traits that were measured in this population. So we began with classical things like height and diameter, but then we moved to more physiological type traits. Using carbon isotope discrimination, we could get measures of carbon and nitrogen, and a surrogate measure of water use efficiency from the C12-C13 ratio. Using another platform, we could get measures of C5, C6 sugar content, lignin content, GC ratio, the things that you would be interested in an improvement of wood chemistry traits. And then finally on the bottom, we did a full metabolomic screen, all the metabolites in wood samples taken from all of these trees. So it's a rather large and exhaustive amount of phenotyping in this population. And by the way, I forgot to mention, but Brian did not forget to mention that all of this work has been done largely by Fernando Guerra while he was working as a postdoc in my lab. That Fernando has returned to his university in Talca, Chile, but continues to work with us on obtaining and analyzing these data. So the phenotyping is all complete in that population. The next step then is to genotype with some type of DNA marker every single clone in the population. There are many opportunities or choices for genotyping these days. This technology advances rapidly. And I'll explain in a minute here the platform that we've used for genotyping in these trees. So then finally in the little panel on the right is just the statistical relationship between the phenotypic value, the dependent variable, and then the genotypic measure to test for association between genotype and phenotype. So now I'm going to move to the next slide, and this is just a summary. Fernando has produced these results. All the phenotypes that were measured in this population. You can see the phenotypes listed in the first column. I would just draw your attention to the minimum maximum values for each of this, the enormous range and variation in that large population. This shouldn't be of any surprise to anyone who works in forest trees, the large amount of genetic variation that exists. And this is important. Without genetic variation, there would be no opportunity to detect the relationship between the genotype and the phenotype. But for every single one of these traits, there's a large amount of genetic variation. And then in the final column, you'll see some estimates of clonal heritabilities. These are all quite encouraging, very modest, if not high, heritabilities, indicating that the high genetic control of these things, again, providing strong encouragement that one could successfully identify marker trait associations. So I'll move to the next slide then. As I said a minute ago, there are many, many different types of genotyping platforms. People would be familiar with the so-called fragment-based technologies that went back many years ago, things like microsatellite markers and so forth. In recent times, we've moved toward DNA sequence-based approaches, and there are many of these as well. But for all of these DNA sequence-based approaches, what you're searching for in terms of genetic variation is what's called SNPs, single nucleotide polymorphisms. That's just a substitution at a single base position for one base for another, an A for a T, if you will. So through DNA sequencing, we can discover hundreds of thousands of these. Because the Poplar Genome was sequenced many years ago, we have the ability to scan across the entire genome, or in this case, the entire gene space. So we're using a technology called sequence capture, exome capture, where we can detect the genetic variation in 40,000 plus genes in the Poplar genome, and that's essentially all the structural coding genes in the Poplar genome. And furthermore, we can find the genetic variation, the SNP variation within each one of those genes. So in that little panel in the lower right, what you would see is each row is an individual, and then each column is an individual SNP within a gene. And what's indicated in that second column, you can just see some individuals are AA, adenine homozygotes. Other individuals are AT, those are heterozygotes, and the other individuals are the alternative homozygote TT. So it's just back to what we all know from Mendelian genetics, 1, 1, 1, 2, 2, 2. But that's done at a very large scale using DNA sequencing technology. So now we have the phenotypic and the genetic information on that same set of 460 odd clones. And now we looked to identify a relationship between those two things. And if we can discover such a relationship, we would conclude that that gene might underlie that complex trait phenotype. And that's where we are in the process of our final year of this project, and where Fernando is right now. We've more or less completed gathering these two very large data sets, the phenotype data set and the genotype data set, and he's analyzing those now to discover those marker trait associations. But what I have in this final slide here is just taken from some earlier work that we did with Brian with the very same population, actually. It says 448 clones, but it's from that same larger population that we're using now. And for some of the same phenotypes, you'll see these wood chemistry phenotypes indicated here on the column on the left. But at this stage of the development of our work, we were only able to work with the genetic variation in a small number of genes, and in fact only 40 genes. Now we're looking at 40,000, so that's 2 orders of magnitude more. But the process is the same of looking for marker trait associations. And we found many. This work was published several years ago in a paper by Jill Wagerson et al. We used 2 types of statistical tests that are indicated at the 2 bullets at the bottom of that left-hand column, a single SNP test or a so-called haplotype-based test. I think the distinction there is not important for this conversation, but what you'll see in these figures on the right is sort of the result of that statistical test. In the upper box there on the y-axis is percent lignin content, and then along the x are the average values for individuals that were of the AA genotype, the AC genotype, or the CC genotype. So that you can see if you're trying to decrease lignin, you might want to select on individuals that are carrying the A allele and not individuals that are collecting the C allele. That's a little bit simplistic, but that's, I think, descriptive of how the molecular breeding process might work. And likewise, you can see for another gene and different SNPs within that gene, the difference between individuals that have inherited 2 copies of the A versus an individual that is a heterozygote, and so forth. So this is the discovery process upon which we can determine that we can discover genetic markers that are predictive of phenotypes. And that's the work that we're doing here at the university to support Brian's program. I think now I can move, yes, I've moved to the next slide, and I think I'm going to be able now to turn it over to Brian to talk about how he and his program would take the discovery knowledge that we've acquired and bring it to application. So if you're ready, Brian, I'm going to go over to mute so we don't echo to the participants.
David, thank you. And to the audience, it should be clear from that presentation of David's and Fernando's that genomic science is really emerging and moving quite rapidly. What's lacking, at least here in the Northwest, is the translation of this science, the application of this science to poplar breeding. It just hasn't happened yet. The work at UC Davis will advance that. But at one time, there was hope that the Poplar Molecular Genetics Cooperative that was led by Toby Bradshaw at the University of Washington in Seattle would be that vehicle that would bring this science, this genomic science, to application in populace. That work was funded largely, that cooperative, the members were largely recruited from the pulp and paper industry, but that industry disintegrated somewhat in the 1990s, along with the disengagement with poplar cultivation in the region. And then that led to the discontinuation of Bradshaw's industrial cooperative. So now here we sit with massive, I'll say, massive genomic resources that are becoming available at a time that we need to account for the translation of this science to applied breeding. And what you're looking at on this slide is a map in the Northwest, Western Washington and Oregon, that shows the provenances that were sampled for the association population that has been the focus of Fernando and David's work. Okay, so in considering then how these tools are going to be applied in breeding, I think it's good to discuss right now the two main obstacles that GreenWood in its poplar breeding program faces. The first challenge is in making an effective prediction of the hybridizing value of any given pure species parent genotype. Now remember, we're dealing with 4 species, Deltoides, Trichocarpa, Nigra, and Maximoitzii. How do we estimate how valuable any one of these genotypes is to the hybridization program? And predicting these values using classical quantitative statistics and field plot trialing is challenging, both in terms of logistics and scale and also reproductive biology. So briefly on logistics and scale, the prediction of breeding value for any one genotype is dependent upon the other species with which it is hybridized in the program. So for instance, in the prediction of a breeding value of the 150 populous Deltoides genotypes, those breeding value estimates have to be made separately in terms of whether it's crossed with Trichocarpa or hybridized with Nigra or hybridized with populus Maximoitzii. So the scale is large. Secondly, predicting hybrid breeding values is encumbered somewhat by the vagaries of hybrid reproduction. It's commonplace that you experience a lack of full reproductive possibility between species, and this complicates, as I'll describe, the estimation of breeding values for all members of the breeding population, both the males and the females. For instance, in the first cross listed there, Nigra deltoides, when populus deltoides is used as the female parent and Nigra as the male parent, the cross is routinely successful. Hybrid progeny are recovered, and there's the plant material available to put in a test plot, design an experiment that will yield quantitative measures of breeding value. In the reciprocal direction, as I'm showing here, the female is always listed first in the cross. In the reciprocal direction, where Nigra is used as the female parent and Deltoides as the male parent, there's a failure in the pollination fertilization mechanism, and the cross fails. So crossbreeding values can be developed then, hybridization values can be developed rather for populus deltoides females, populus nigra males, but not for Nigra females nor deltoid males. A similar phenomenon is commonplace in the next cross, the generosa cross, where crosses between Trichocarpa as the female and Deltoides as the male, normally we see a premature abortion of the capsules, the seed capsules. And we realize or fail to realize a recovery of hybrid offspring for experimentation, once again, to recover plant material that is the basis of an experiment to extract quantitative estimates of breeding value. So what do we do? Now, we're running a little late on our webinar. This is the point in the webinar where we would go to the question and answer session. So because of the technical difficulties, we're running behind. I'm going to skip over the second challenge and go right to the plan that GreenWood has in place to use the information being developed in David's lab to circumvent this problem of logistics and scale and reproductive biology that precludes the estimate of hybrid breeding values via the normal classical approach of quantitative statistics. So what do? We have these 4 species. We would like to extract for each of the 150 genotypes within each of the breeding populations. We'd like to extract hybrid breeding values to guide our hybridization program. So what the plan is now is to employ computational genomics, and we will use this to, we don't, let me mention that we are not in the AHV program developing prediction models. We're only developing the association statistics. We're not developing the prediction models. Those prediction models, the models that will use the information that David and Fernando are developing to predict directly these genomic-based breeding values, we don't have those models yet. So what we are doing in the interim is considering an indirect approach. I'll describe it as breeding population management, under which we'll take our generation, for the Generosa program, as an example I'm showing here, we are going to simply identify which of the Trichocarpa and which of the deltoidy parents represent the most unique genetic haplotypes in the respective collections. That is, which are the parental genotypes that contribute the real rare haplotypes to the breeding population pool when it's brought together in a hybrid cross. And what our expectation is that when the F1 generation is recreated using the most unique set of haplotypes of each of the 2 species, what we will do is recover a distribution of hybrid family genetic values that doesn't necessarily follow a normal distribution that we often see, but rather, because these F1 families are going to be bred from the most diverse or unique set of haplotypes, what we are expecting is a bimodal distribution in the F1 generation that includes, at the right-hand side of the distribution, the high-value families. And what this should do is, maximize the efficiency with which selections from unique elite families are going to be recovered from our testing program. So again, we're not using genomic-based breeding values yet. We're not in the position, because we don't have the prediction models yet, to guide our breeding program. Rather, what we are going to do is use the genomic information simply to identify, by scanning our population, where in the population do the unique haplotypes reside, use those unique haplotypes in recreating the first generation. And what this will do, we believe, our hypothesis is, this will lead to an increased recovery of elite families. So we're going to test this hypothesis. We'll haplotype our breeding populations. We'll design a crossing program that includes hybridization of the common as well as the rare haplotypes. Then we'll conduct a field trial to test the performance of the families as a function of haplotype diversity. Okay, again, apologies, but in the interest of time, I'm going to skip over the second translational approach, which follows more of the traditional, not traditional, but rather the common genome-wide selection protocol that most people are talking about, and go directly to a conclusion of our seminar, leaving about 10 minutes for question and answers. So I do want to mention that the one thing we've learned from those demonstration test sites that I showed at the outset of the presentation is the importance of varietal selection and maximizing biomass production at each of the sites. And to capitalize on the GreenWood Resources breeding program that has bred these varieties and can design to improve yields, but now we're going to be shifting emphasis to traits that heretofore were not considered in breeding these varieties that are under test. They were bred mainly for the pulp and paper market, but now we're going to be redesigning our breeding program to select for such things as coppicing ability, carbohydrate content, some of the physiological measures, drought use or water use efficiency and nutrient use efficiency that David talked about, growth performance and high density plantings. To do this, new tools are needed to keep the pace of genetic improvement going. The AHB project, David and Fernando, they've are realizing very good success in the association study, and I'm quite excited about this first step that I've described about taking this information, this science, to the translational stage. So thank you very much on David's behalf, my behalf, for your attention. I will now, I think, turn it over back to you, Patricia, to moderate questions and answers.
Yes, thank you, Brian, and thank you to our participants today for being patient with our technical difficulty. I'm glad you hung in there and were able to hear David's part of the presentation. So please type your questions into the chat box. We will having another webinar on Poplar on February 25th at 10 a.m. Pacific time, where we will have Bruce Summers of GreenWood Resources, and he will be talking about harvesting short rotation poplar for bioenergy. We will also have John Poselius from Payson Holland will join him as well in that webinar. As you're typing in your questions in the chat box, if you would also look at the poll questions we have on the right side of the screen, if you could answer those as well. And the way we're going to be handling questions is that Brian is going to try and answer the majority of them. And then when there is a question that is better for David to answer, Brian will field that question to David. So I'm going to turn the mic back over to you, Brian, and thank you for joining us today in this webinar, and thanks to USDA NEPA for providing funding so that we can have this webinar series.
Patricia, thank you. So our first question comes from Rick Hall, Iowa State University. Do we anticipate yield improvement from engineered sterility in addition to other advantages it would provide? So the project, the AHB project, does include work at Oregon State University, led by Steve Strauss, to engineer sterility into varieties of poplar as an enabling technology that would promote the commercial deployment of varieties that are engineered for traits of economic importance. So the sterility there has value as an enabling technology. I do not necessarily feel that it would directly contribute also to an enhancement of yield if the varieties were made sterile. It may, you would think, if you shunt away from reproductive growth into biomass growth, you would see a yield enhancement. But the majority of the varieties that have been selected, our most productive varieties, are female, that I think have a higher component of photosynthase shunted to reproductive growth than their male counterparts. Nonetheless, they are some of our higher yielding varieties. So we may see that, but I'm not too overly optimistic. So the second question comes from Judd Isobrands. Have you standardized within tree trait sample collection? We have for such traits as wood-specific gravity, stem taper. I don't think we've standardized it yet for our leaf analyses of nutrient use efficiency or water use efficiency. We're following the same protocol that David has used. We do have a proof of concept trial underway right now at one of our farms, where we are comparing hybrids that have rated very high in water use efficiency based on stable isotope discrimination. We are taking those as well as counterparts that have not rated so highly in water use efficiency, and they have been placed in a factorial trial, the second factor being the rate of irrigation, to see if these water use efficient and less water use efficient varieties respond to varying levels of irrigation as we would expect them to. So that's in process. We are standardizing that.
Hey, Brian, I said I wouldn't jump in unless prompted, but maybe, can you hear me?
Very well, David.
Yeah, I might have interpreted the question a little bit different in regards to actually the sampling that we conducted in the field for these tissues that were then subsequently phenotyped because of the large amount of environmental variation that might occur if sampling wasn't standardized. I think possibly that's what Judd was alluding to. And what I will just say, he can read later in publications, exhaustively, for these reasons, that leaves were taken from the precise locations in the crown among all trees, increment cores were taken from the exact location, the same annual ring was used to sort of minimize that environmental variation if you did not standardize. So I'll go off here and go back to mute, Brian.
David, thank you for that clarification. Good clarification. Third question is from Bernie McMahon at the University of Minnesota. Any hope for a national approach to development of association populations in the important species beyond the Pacific Northwest, including the Midwest, the Southern U.S.? Does the University of California, Davis work translate across populations? So, yes, I think under the Sun Grant Partnership Program funded by the Department of Energy, that includes the University of Minnesota, GreenWood, Arborgen, Michigan State University, and the University of, or Mississippi State University, there is an effort to collaborate among all institutions to bring together association populations for experimentation and development of these genomic resources. David, the second part of the question, how do you see your work translating across different species populations? Can you answer that?
Yeah, the answer to that will come when we do it. Get into this discussion all the time. I would argue that for some marker by trait associations, those are going to be very highly conserved from one species to another. Those are very important pathways that are ancestral to the taxa. But for other things that we may see that different species have evolved different strategies and that different genes are being recruited to determine phenotype. So both. And it's important to understand what both are. So the work that we've done with tricho really needs to be repeated in all the other parental species of this hybrid program. And we would love to see that work could be done ultimately.
Okay, I think we're coming up to the hour, but at least I will stay on. It seems that there may be additional questions coming. I can stay on. I don't know if David is also, but let me take the next question from Donal. Have I got it right in thinking that in David's presentation that each clone is subject to DNA sequencing? Sequencing each of the more than 40,000 genes in all of the 400 plus clones. David?
Yes, that's absolutely correct. And unfortunately, Brian, it would be appropriate for me to stay on the call and answer questions here, but there's a group waiting for me to lead a discussion in another room. So I'll maybe try to hang
Okay.
on for just a minute or two, but I am going to have to leave for another gathering.
Understood. Okay, Patricia, it looks like there are no more questions coming in. Will you conclude us? Oh no, I see Bernie McMahon is typing one.
So Brian and David, thank you for a very informational webinar. Thank you for our participants for being patient today. And all of you have a good day. As I mentioned in the chat box, this webinar was recorded and will be located on the Hardwood Biofuels website. I just noticed I typed in biofuels instead of biofuels, but you can find the webinar there. So thank you much for a good day, and we hope to have you present on the next webinar on February 25th, 2015. All right, bye-bye.
Thank you.